Welcome to this fact sheet video on rigorous methods for stem cell-based models of neuropathic pain. My name is Pascal Röderer, and in this presentation I would like to walk you through some key resources, differentiation strategies and practical tips for working with iPSC-derived models in the context of neuropathic pain research. Recently, the International Society for Stem Cell Research published standards for human stem cell use in research, as well as recommendations for the reporting and publishing of results obtained with human pluripotent stem cells. You can find all these information on the ISSCR website. To begin, it's important to know that there's already a rich global infrastructure supporting iPSC-based research. Several national and international stem cell repositories provide access to well-characterized cell lines. Examples include the European Bank for Induced Pluripotent Stem Cells, the African iPSC Initiative, or the New York Stem Cell Foundation. Turning now to sensory neuron differentiation. We typically start from tissue donors, for example, providing PBMCs or fibroblasts. These somatic cells are reprogrammed to induce pluripotent stem cells using the so-called Yamanaka or reprogramming factors OCT4, SOX2, c-MYC and KLF4. Sensory neurons can be generated from iPSCs employing different strategies. Small molecules can be used to directly guide iPSCs into a sensory neuron fate, and I will talk about this a bit more in a minute. Alternatively, overexpression of fate-specifying transcription factors can be used to quickly generate neurons. Other protocols employ the intermediate generation of a stable population of neuronal progenitor or neural crest cells. And of course, these three approaches can also be combined. Now we want to focus on the small molecule approach. One influential protocol published by Chambers and colleagues in 2012 describes a small molecule-based approach to generate sensory neurons from pluripotent stem cells. To date, this is the most used and modified protocol for the generation of sensory neurons from human iPSCs. Here, pluripotent stem cells are first driven towards a transient neural crest cell identity by passing alternative fates such as trophectoderm, mesendoderm or non-neuronal ectoderm, before then initiating a sensory neuron identity. This fate decision is achieved by carefully modulating signaling pathways including TGF-beta, Notch, FGF and Wind using different small molecules. Subsequently, exposure to neurotrophic factors such as NGF, GDNF and BDNF supports the transition from immature to mature sensory neurons. Now, I want to briefly go through the differentiation process with you step by step. Here you can see the timeline of a recent variation of the Chambers protocol. I will want to focus especially on the first 10 days, in which the iPSC transition towards a post-mitotic neuronal identity. Starting with day in vitro 0, you can see how iPSCs look like after seeding at different densities, ranging from 50 to 200,000 cells per square centimeter. Differentiation is then initiated by a media change to differentiation medium supplemented with small molecules for dual SMART inhibition. Cells proliferate and grow more dense. Especially with higher initial seeding densities, you will see the plate is quickly confluent. Starting with day in vitro 4, small molecules for the modulation of Wind, Notch and FGF signaling are added to the medium. Day 6 of differentiation is an important time point where usually a lot of cells start to die and it looks like the dense cell layer breaks open. Now you can observe that the cells that survive the cell death of day 6 form little clusters from where neuroids start to grow and neurons start to migrate out. On day 10, the plate should be covered in neuronal clusters and a dense network of neuroids becomes visible. And when we zoom in, we can clearly see that the 200K condition produced a relatively pure neuronal culture, while the 50K condition was not optimal and produced also a lot of non-neuronal cells, as can be seen here in the middle. Differentiation doesn't stop here. neuronal development continues over several weeks in vitro. The morphological and functional maturation of iPSC-derived sensory neurons is a dynamic, time-dependent process. Allowing sufficient time in culture can be particularly important when modeling chronic neuropathic pain phenotypes, which may depend on the expression of ion channels and receptors that only come up after prolonged maturation. Once cultures are established, several complementary tools can be used to rigorously characterize and analyze iPSC-derived sensory neurons. And here we summarize some examples. First, calcium imaging allows us to monitor activity in large populations of neurons and assess responses to stimuli in a relatively high throughput way. Second, immunostaining enables us to verify neuronal and sensory marker expression, for example, staining for peripherin with brain3A or sodium channels to confirm neuronal identity and culture quality. Third, patch clamp electrophysiology provides detailed information on membrane properties, action potential firing, and ion channel function, as illustrated by the current clamp traces on the slide. Last but not least, multi-electrode array recordings can be used to get an insight on the population activity of sensory neuron cultures. At the end, I would like to summarize some practical recommendations for the work with induced pluripotent stem cells. First, always maintain high-quality iPSC lines and regularly check for contamination, pluripotency marker expression, and genomic integrity. Second, get to know your cell lines. In particular, how they respond to different initial seeding densities, since this often determines whether differentiation will succeed. Third, optimize culture conditions for your specific assay, including cell density and coating matrix. There is no single set of conditions that works best for all readouts. Finally, remember that development in vitro is an ongoing process. Systematically monitor both the expression and functionality of your targets over time, rather than relying on a single time point. Thank you for your attention, and a big thank you to the whole team that helped writing the fact sheet and producing this video, as well as the co-chairs of the Global Year on Neuropathic Pain, Angelika Lampert and Neil O'Connell. For more information on neuropathic pain and access to the full set of fact sheets and resources, please visit the IASP website.